Okay, and an official good evening to everyone. Welcome y'all to the third edition of the 2026 Naturalist Webinar Series. My name is Dana Ripper. I'm with the Missouri River Bird Observatory, or MRBO. And also with us tonight is my co-founder and co-director, Ethan Duke. Here at MRBO, I think lots of folks know our mission is the conservation of birds and all wildlife and their habitats through science, education, and advocacy. And we definitely mean all wildlife, as I hope you are learning from this series. As I usually do in these introductions, I'd like to note a few of our upcoming events. This Thursday evening, February 19th, I'm going to be giving a presentation with MRBO's awesome longtime education partner, the Daniel Boone Regional Library of Columbia, but it is online. I will put that registration link in the chat, and the topic is wintering raptors in Missouri. And then if you happen to be in central Missouri, our excellent educator, Melina Matheny, is giving a bird feather lab program for kids at the Marshall Public Library on Thursday, February 26th at 5:30 p.m. Students that attend, they'll be engaging in hands-on activities while learning about how feather structure helps birds fly, swim, and stay warm. This is one where it's in person and you can just show up, but I'm going to put the information in the chat so that you can copy it if you like. I'm going to go ahead and do that right now. Here is the wintering raptors one. Nope, that didn't do a link. I will do it later. Sorry, y'all. But here is the bird feather lab one. This is not going to be a link anyway. This is just the information about it. And please do know, of course, that all of our upcoming events, both virtual and in person, are always on our website at mrbo.org. We have a whole calendar, and the MRBO team is always out and about giving various programs, and you are most welcome to join us. My final note here, we are recording all of these webinars, and they will be on the MRBO YouTube channel, along with literally at this point, hundreds of others. And that I will put in the chat as a link. And so you can always catch webinars there and share them with other people if you would. So on to our webinar of the evening. We are delighted to welcome Professor Waylon Hiler of Missouri Valley College to speak with us. Waylon holds an MS degree in biology from Arkansas State University, where he studied the Ozark hellbender. He is an associate professor of biology at Missouri Valley College in Marshall, where he has been teaching since 2008. Previously, he was a project manager and biologist at TRC Environmental Corporation in Laramie, Wyoming. His research interests focus on the ecology and conservation of amphibians and non-game fishes. In addition, he has spent the past 9 summers working with Colorado Parks and Wildlife on a long-term research project that examines the cause-specific mortality of neonate elk in the Rocky Mountains. He has over 26 years of field experience in environmental data collection. Tonight, Professor Hiler will be speaking to us about Missouri amphibians, notes on their natural history, diversity, and conservation. So Waylon, the floor is all yours.
No, thank you, Dana and Ethan. It's a pleasure to be here. I thought I would start out tonight's presentation talking a little bit about my background, how I got to where I'm at, a little bit of what I do at Missouri Valley and my teaching philosophy, and then get into the main subject, which is amphibians in Missouri. One of the things that I find myself doing before I go into the field, whether it be for vacation, maybe down to the tropics and doing some snorkeling or scuba diving on a coral reef, is prepping myself with a little bit of natural history and diversity work, so I know what I'm looking at when I'm in the field. And so I thought I would spend a little time this evening talking about the natural history of amphibians, some general characteristics of amphibians, and then get into some conservation issues that our local amphibians are facing here in Missouri, and with particular emphasis on the hellbender. And so that's, as Dana mentioned, that's my background in terms of my graduate research. So on the slide, we have a gray tree frog. I love this photo just because, it's a great example of how closely the color pattern of some of our amphibians match their background environment. So the gray tree frog is perfectly camouflaged and cryptically colored to match the bark of the trees that it rests on. And this photo was taken at Van Meter State Park. As Dana mentioned in my introduction, I've spent the last 8 or 9 years doing field work for the Colorado Parks and Wildlife as a crew lead and research assistant for a neonate survivorship study taking place in the heart of the Rocky Mountains. And I included this photo because I think it, first off, is a spectacular photo of a newborn elk calf with Capitol Peak in the background, inside the Maroon Bells Wilderness Snowmass Wilderness area. However, I am truly a field scientist. I didn't set out along this path that I'm on thinking that I was going to be in academia. I imagined myself working for a state wildlife agency in Colorado. That's where I'm from. And, and life is unpredictable. When I came to college, I was mentored by a herpetologist, Mark Mills, who's now at Missouri Western. And he offered me an opportunity to do research on snapping turtles at a local state park. And so I took that opportunity, and it one thing leads to another. And eventually I found myself working with hellbenders focused on amphibian conservation. But at Missouri Valley, we're a teaching institution. And so I'm able to spend my summers, doing stuff that I feel is relevant, that is purposeful, and at the same time kind of feeds my interest as a general ecologist and someone that is passionate about the outdoors. Because I think that's what attracts a lot of us to biology and conservation, is a love for the outdoors and not just for one faunal group or floral group. And so this is what I've got to do the past 8 or 9 years in the summertime. At Missouri Valley, we teach a series of natural history courses that I enjoy teaching. So ichthyology, herpetology, mammalogy. My teaching philosophy is centered around outdoor education, hands-on learning experiences. Those are the types of things that stuck with me. When I was a student, when I was kind of finding my way as a biologist, I really remember the field trips, the camaraderie that's built between classmates, getting to interact with your professors on another level, and then ultimately getting to observe things with in the field that you learned about in the classroom. So it's a nice connection. And so we take students to Rice Biological Station, which is outside Steelville, operated by St. Louis University, but we're fortunate enough to have access to it. So Professor Michelle Ranke and I, Michelle teaches the ornithology class and the wildlife management and ecology classes here at Missouri Valley. We lead joint field trips to Rice every semester in hopes of making a difference in our students' lives, because not all of our students are going to become professional biologists, but it's an important opportunity to hopefully grow their appreciation for conservation and nature. And a lot of the photos that I'm going to show tonight came from Rice. The Ozark Plateau, which is in our backyard here in Missouri, some of you might be in the Ozarks right now, are a biodiversity hotspot for salamanders. And I'll talk a little bit about that as I work through my presentation, but it's just a spectacular place to go and experience unique biodiversity. As I mentioned, the teaching philosophy that I focus on is experiential learning, hands-on learning. So not just Rice, but we lead field trips to Utah every spring. This is a photo from a group of students and I in Belize. And so the hands-on learning and just getting people acclimated to the outdoors, I think is an important process of their education. And then I've also been fortunate enough to work with some of my former students. And so this is Zach Durbin. He's a conservation agent for the Missouri Department of Conservation. And he spent 2 years working with the Colorado Parks and Wildlife on the neonate study. And then he also did some lion work, some mountain lion work, and mule deer work. So he is one of our all-stars at Missouri Valley, and we're proud of him. All right, getting into the meat of the presentation. So before I talk about some general characteristics of amphibians, I thought I'd take just a few moments to talk a little bit about the evolutionary history of amphibians. Obviously, vertebrate life originated in the oceans, and our first tetrapods, so our four-limbed vertebrates that made their way out of the oceans, stepped on land around 260 million years ago. And those first tetrapods were very similar to what we might imagine, an ancient amphibian to be like. There were some ancestral characteristics they carried with them out of the water, scales on the belly, a few other features, but eventually those aquatic features were lost, and a more terrestrial tetrapod evolved. Now, early in tetrapod evolution, there was a split between amniotes and non-amniotes, and amphibians are non-amniotes. I'm going to talk a little bit about what that means here in a moment, but it was a major division. One lineage gave rise to our modern amphibians, and then the other lineage gave rise to reptiles, birds, and mammals. And so on the cladogram that we're looking at, I have a few points highlighted, the common ancestor of all of our modern tetrapods. The temnospondyls were ancient amphibians that gave rise to our modern clades of amphibian. And, our current taxa, the caecilians, which belong in the Gymnophiona, the Anura, which include our frogs, and the Caudata, which include our salamanders, share a common ancestor somewhere around 240 to 275 million years ago in the Permian period. So this is preceding the Triassic period. So amphibians have been around for a really long time. Amphibians that you would recognize have been around for a really long time. So if we look at the kind of the breakup of Pangea, just a kind of a brief synopsis of the breakup of Pangea and continental drift, you can see that, the common ancestor of modern amphibians had already evolved prior to the breakup of Pangea. So some of the interesting distributions that we see in our modern amphibians, they elicit questions. Why do we have, hellbenders in North America? And then we have, these close relatives, one in Japan and one in China and nowhere else. Why are plethodontids the only salamanders in the southern hemisphere? So when we start asking those types of questions, we need to come back to this idea that plate tectonics has played a critical role in the biogeography of our modern amphibians. And then also with warming and cooling periods, throughout Earth's history, their distributions have been shaped by glacier movements, land bridges forming and then disappearing. And so I don't want to, with only 45 minutes, available tonight, we can't spend too much time talking about the biogeography of amphibians, but it is an interesting thing to consider when we start studying these different groups or clades of amphibians. Now, salamanders are not quite as diverse as frogs. I'm not going to speak, on caecilians tonight. We don't have any of the limbless amphibians represented here in North America, and so I'm not going to spend too much time talking about them. The focus of tonight is Missouri. But globally, there are about 619 species of salamander spread throughout 10 different families. 40% of our extant taxa, though, are threatened with extinction, or at least imperiled, meaning that they are threatened with decline. And that's alarming. I am going to talk a little bit about amphibian decline tonight, specifically hellbender decline. But also a little bit as to why our amphibians are so imperiled as a group. In Missouri, we have 7 families and 20 species of salamander, which doesn't seem like a lot until you compare it to the overall global diversity. And we have 10 families globally, and 7 of the 10 families are represented here in Missouri. So we are truly in a biodiversity hotspot for salamanders. We can see a wide range of different taxa. So the Ozarks have the second highest amount of biodiversity in terms of salamanders, and then the Appalachians, a little bit further east, have the highest amount of salamander diversity in the world. And so again, another cladogram illustrating the relationships between our salamander families. The ones highlighted in pink are families that we have in Missouri: the plethodontids, the lungless salamanders, amphiumas. They're eel-like salamanders, are mud puppies, pedomorphic salamanders that retain larval characteristics. I'll show you an image of a mud puppy in a little bit. Are mole salamanders in the family Ambystomatidae. This is between the plethodontids and the Ambystomatids. These are probably the 2 families that most people outside the herpetology community are familiar with. Salamandridae includes our newts, which have a really interesting life cycle. They undergo 2 metamorphoses. Cryptobranchidae are giant salamanders, and then our sirens, the most ancestral salamander family that we have on Earth today. And then also, the one thing I like about this cladogram is that it illustrates the divergence points for our different families. And so shortly after the salamanders and frogs diverged from one another, we have, a radiation that produces the cryptobranchids and the hynobiids. The hynobiids are Asiatic salamanders, but the hynobiids, the cryptobranchids, and the sirens are all ancestral. They all fertilize eggs externally, which is different from the rest of the salamanders. And I'll talk a little bit about salamander reproduction in a few slides. And so we have this early split from the sirens, and then we have another split that produces 2 ancestral lineages. And then we get into kind of the meat of salamander diversity today, and Plethodontidae is by far the most speciose family of salamanders. Frogs are more diverse, more species, 48 families globally, around 6,200 species. However, new species of frogs are being described, and so as we explore new habitats, or start doing genetic analyses on different lineages, we realize that we have cryptic species that we need to split into multiple species, or we discover new species. And so the number is growing, above 6,200. However, 30% of all frogs are threatened with extinction. And then another 25% on top of that are we know so little about, we can't provide an accurate population assessment. And so there's a lot of unknown with a quarter of the frogs that exist on Earth, and then over a third of the frogs that exist on Earth are threatened with extinction. In Missouri, we have 5 families, 25 species. Relative to overall diversity, it's not as high compared to the salamanders. Again, 7 out of 10 families of salamanders represented in Missouri. And I think when most people think of amphibian diversity, their mind would take them to the tropics, and that would be accurate if we're thinking about frogs. However, if we're thinking about salamanders, we need to think northern hemisphere, kind of subtropical and temperate latitudes. This is the wood frog. I included this photo. I chose the wood frog, I guess, because it has such a unique life history. Wood frogs are freeze tolerant, or meaning they can basically overwinter in areas above the frost line. So they don't have to get below the frost line to survive a winter. They are adapted to northern latitudes. This is a subarctic species. The northern temperate latitudes that, if we're thinking about Canada, southern Canada down into the northern part of the United States, that's the optimal habitat. But during the last glaciation period, glaciers moving south, advancing down the North American continent, pushed floral communities south, faunal communities south, and then as the glaciers retreated, we have these relic species. And so the wood frog is one of those relic species, hence its freeze tolerance. The four-toed salamander is also a glacial relic from the last ice age. And so the populations of wood frogs and four-toed salamanders that we have in Missouri, again, are courtesy of the last ice age. In terms of how a wood frog is able to survive freezing temperatures, there's some physiological mechanisms built into its internal anatomy, but water is evacuated from cells, and then cells are filled with kind of an antifreeze compound produced in the liver, and removing the water from the cells, filling it with an antifreeze prevents the cells from rupturing and bursting. And as temperatures warm in the spring and their opportunity to reproduce approaches, they'll unthaw and become active. So it's a really extraordinary species that we have right here in our backyard. This photo is taken at Elephant Rock State Park in the Ozarks, and so this is a Missouri wood frog. And then we're looking at the distribution of wood frogs here, and you can see that in Missouri we have, kind of these disjunct populations, and even further south into Alabama, we have some wood frogs that were pushed south by the glacial advance. So some general characteristics of amphibians. Amphibians are non-amniotes or anamniotic. And what that means is that their reproduction is tied to water. So their life cycle is tied to water, not just reproduction, but and I'll talk about it a little bit more here in a moment. But when we think of amphibians, we should think of water, and their reproductive cycle is tied to water. And so their embryos are not encased in a water compartment like reptiles, birds, and mammals encase their embryos. So our embryos are encased in an amnion, which is a fluid-filled sac, and that fluid-filled sac keeps the embryo from drying or desiccating as they're developing. And it makes sense in terms of kind of the progression of vertebrate evolution. Amphibians evolved from the first tetrapods. Tetrapods evolved from our lobe-finned fishes. Obviously, fish reproduction happens in water. Amphibian reproduction remains tied to water today. The other lineage of tetrapods, the amniotes, evolved these extra embryonic membranes that protect their embryos, keep them from drying out. And so right away, if we're trying to describe the difference between a salamander and a lizard to maybe someone that is curious about what we learned tonight, or maybe we're talking to a child that we want to take into the woods, and they ask us, what is the difference between a salamander and a lizard? We can start with that. Their reproduction is tied to water. So reptiles reproduce independent of water bodies, whereas amphibians need water for reproduction. This is a salamander egg mass, a spotted salamander egg mass. So some amphibians lay large masses of eggs that obviously swell as they are oviposited into the environment. One tiny little salamander couldn't fit all those eggs in her body as they are right now, but kind of the gelatinous mass that surrounds the eggs obviously absorbs a little bit of water. So some amphibians reproduce in standing water, like the spotted salamanders. Some amphibians reproduce in flowing water, like our stream salamanders, desmogs, and hellbenders, for instance. Some amphibians reproduce in just moist microenvironments, and I'll show you some images of that taking place tonight also. Salamanders have smooth skin. So this is a dark-sided salamander. You can see how shiny it is. The skin stays moist because of mucus glands in the dermis, and the moisture is required. Well, not only the mucus glands help supplement the moisture, but the amphibians tend to live in moist and wet places, which keeps their skin wet. But the moisture is important to facilitate gas exchange with the environment. So not only do amphibians use lungs for respiration, they also practice cutaneous respiration, which is just exchanging gases through the epidermis directly into blood vessels embedded in the dermis. And then at certain points in their life cycles, some amphibians have gills. And so they utilize 3 different respiratory surfaces to obtain oxygen. Now, this is a histological section of amphibian skin. And the one thing I want us to look at tonight is the upper layer or the outer layer of the integument. So just in this bracket on the top right, it says epidermis. And then everything below the epidermis belongs in the dermis. And so vertebrate integument is stratified into these layers consistently, regardless if we're talking about fish, amphibians, reptiles, birds, and mammals. They all have an epidermis, which is essentially a layer of keratinized cells, which acts as a barrier against pathogens in the environment, protects against physical abrasion. In our amniote groups, the epidermis helps in water conservation, and it's a little bit thicker and it contains more keratinized cells. Amphibians have a very, very thin epidermis, and that thin epidermis is required if gas is going to be exchanged between the environment on the outside of the epidermis and the blood vessels that run through the dermis. And so not only are they exchanging gases with the environment through their epidermis, the thin epidermis, but they're also absorbing water. So they're osmoregulating through their skin. And one of the things that we're going to talk about in terms of conservation issues with amphibians is chytridiomycosis, or the pathogenic fungus that we just call chytrid, and it's in Missouri. But one of the issues with chytrid is that it thickens the epidermis and it inhibits gas exchange. It interferes with osmoregulation, and those types of disruptions impact other aspects of the amphibian's physiology, which in some groups is lethal. Other amphibian groups tend to tolerate the infection and sometimes clear the infection. So there's a lot of unknown with chytrid. I'll talk a little bit about it later. But this thin epidermis is characteristic of all amphibians. So all amphibians have this thin epidermis. The SG, that's the layer of cells, the stratum germinativum, where mitosis is taking place. And so if you think back to your high school biology and you're learning about cellular division, these are where the cells are dividing and then migrating towards the surface of the epidermis. And in the process, they're keratinized. And keratin is a protein. It's a structural protein. It's the same thing in your fingernails, in the hooves of a horse, or the claws of your dog or cat. And it's just really, really thin in amphibians. Now, a couple other interesting aspects of the integument. This gland right here labeled MG is a mucus gland. And so amphibians have a dermis filled with mucus glands that secrete mucus onto the surface of the skin through a duct. And so imagine just a little tube running from the mucus gland to the surface of the skin. If the skin begins to dry out, mucus can be secreted onto the skin to help keep it moist. If it dries out, that's an issue, because then all of a sudden the osmoregulation stops and the gas exchange stops. And then the other structure that I think is really interesting is labeled PG right here in the middle, poison gland or granular gland. These are an amphibian's first line of defense. This really almost their only line of defense against predators. All amphibians have not only mucus glands, but a series of poison glands in their dermis that acts as a predator deterrent. We don't have anything in Missouri that is so toxic that it could prove lethal for a human. At the most, it's a mild irritant. In North America, we do have some species that produce toxins that could be lethal. So the rough-skinned newts in California on the West Coast produce a serious toxin that could, in one individual, could kill an individual or kill multiple humans, Also poison dart frogs in South America The dendrobatids are well known for the toxicity of their poison that they produce inside their granular glands. One thing that's interesting about amphibian poison is that it varies wildly between groups, and so the way that the poison acts on another organism varies between group. There's quite a bit of variability. And also, for instance, the dendrobatids, the poison dart frogs in South America, their poison, the toxicity of their poison is enhanced by their diet. And so the arthropods, the insects that they eat, they tend to sequester compounds from their bodies and then incorporate it into their poison, which makes it especially toxic. Now, not all amphibians have smooth, shiny skin. So toads as a family they have a thicker epidermis, and so their skin is a little bit thicker. They tend to tolerate drier conditions, so it's not completely uncommon to be on an upland, in an upland habitat, maybe on the side of a hill, and there's not quite the moisture that you would expect if you were looking for, frogs or salamanders, but you might come across a toad, which is a type of frog. That's possible because they have a thicker epidermis. They're a little bit, their natural history is a little bit more independent, I should say, than other frogs because of that thick epidermis. Their skin is warty, so you see the bumps on the back of the frog. This is a Woodhouse's toad. Those are clusters of granular glands, and so they have these clusters of granular glands, a little bit more concentrated throughout their body, and then the large glands behind the eyes are called parotoid glands, and again, this is a conglomeration of granular glands. So you don't see parotoid glands in other frogs outside the family Bufonidae. You typically don't see this warty skin in other families of frog. So just some characteristics of one of our more common families of frog here in Missouri, the Bufonids. This little circle on the side of a frog's head is called the tympanum, essentially their eardrum, which is exposed directly to the environment. And so they hear fairly well, and it makes sense because they vocalize. The males vocalize a lot, and so if they're vocalizing, they have to be able to receive those sounds. It's an important aspect of their reproductive biology, and so the tympanum is right there. So some frogs, like our bullfrogs, have gigantic tympanums. Other frogs are a little bit more inconspicuous. The skeletal anatomy of frogs and salamanders is also interesting. Salamanders have an elongated body. They have 4 toes on their forelimbs and 5 toes on their hind limbs. They can regrow their toes in some cases. So some groups of salamanders could have their toes clipped or lost and then eventually regrow their phalanges, which has all kinds of applications to humans. If we could figure out how they do that and apply it to, our own bodies, that would be significant. Frogs, however, have a highly modified skeletal system. Their vertebral columns are shortened. Their vertebrae are firmly articulated, so there's not a lot of bendiness or flexion in the vertebral column. So their bodies are compact. So these long legs that are elastic with their muscles and tendons and elongated as they jump and propel themselves forward, you have a nice compact body, which increases the distance of a jump. And some frogs can jump, 30 times their own total length. So their entire body is centered around this type of jumping locomotion or saltatorial locomotion. Another thing we might notice is that they do not have a tail. Frogs are tailless, and that's where the name of their clade anura comes from. Ura referring to tail and lack thereof. So there are tailless amphibians, and so they don't have any caudal vertebrae, the vertebrae that are posterior to the pelvis. However, those caudal vertebrae have been modified into a special structure in the pelvic girdle called the urostyle. And so as frogs are jumpers, they have the hind legs to propel themselves, And when they land, it's quite an impact. And so having this basket-shaped pelvic girdle helps absorb some of the shock, the mechanical shock from landing. And so the Eurostyle is an adaptation that is unique to our frogs. In a few other adaptations in terms of relation to jumping, the intermediate elements of their limbs are fused, and so the radius and the ulna are fused into a radio-ulna, and then the intermediate elements in the hind legs have been fused into a tibiofibula. So the two bones have essentially fused into one, which increases the sturdiness of the limb, because a bone break could be a serious issue for a frog. Amphibians have teeth. That's another question I sometimes get. Do salamanders have teeth? Do frogs have teeth? They absolutely have teeth. Their teeth are pedicellate, meaning they have a structure that's rooted in the dentary in terms of our salamanders. The dentaries are the lower jaw bones. And so they have a pedestal rooted in the dentary, and then they have a crown or a cap that is attached to the pedestal with soft tissue, fibrous connective tissue. So the crowns of the teeth are somewhat flexible, and they can be replaced if they break off. Frogs also have teeth, but the teeth are limited to the upper jaw. They don't have teeth in their lower jaw, but same basic construction, this pedicellate dentition. This is an Andreas japonicus, and I'm wearing a white glove, and because I'm bleeding, because the previous salamander that we were taking measurements on did bite me. And so again, they have teeth, and they're fairly sharp teeth, but I'm still happy. I was still a happy camper in that photo. Getting into the reproduction of salamanders, one of the structures that I think it's important for us to understand is the cloaca. So, as mammals, we're especially humans, we're familiar with our own reproductive anatomy. Genitalia in males held outside the body, internal fertilization. An embryo develops inside the uterus after fertilization in the oviduct. But it's a little bit different in amphibians. So let's just start at the beginning. So amphibians have what's called the cloaca. A cloaca is an internal sinus that the urinary system, the digestive system, and then the reproductive systems all empty into before products are excreted or secreted into the environment. And so the intestine, the digestive system, the urinary duct or ureter running into the cloaca, and then a genital duct running into this internal sinus. And then this vent is the opening to the external environment. So when we think about how fertilization takes place, there's no intermittent organ in amphibians. So that requires either external fertilization, like fishes, meaning the females deposit eggs in the environment and then the male spreads sperm over the eggs, or if we're doing internal fertilization, there has to be some sort of specialized structure. Now, all of our frogs in Missouri are external fertilizers. Eggs deposited in the water are fertilized. It's haphazard. If you've ever been in the woods, during a breeding event, sometimes there'll be a female and there might be 4 or 5 males trying to unplex or grab, any part of her body so they can be in position to fertilize some of the eggs that she deposits in the environment. This is the prototypical position where a female is grabbed or amplexed by a male. And the male is just there making sure that, his cloaca is positioned next to her cloaca. So as she releases eggs into the environment, he's the one responsible for fertilizing those eggs. Now, this was the best photo I had of frogs and amplexus. These two, this is a photo that we took in Utah on one of our field biology trips. So we don't have the red spotted toads in Missouri, but it illustrates amplexus. Now, all of our Missouri frogs produce tadpoles. If we're thinking about how the eggs develop or how the embryos develop, this is typical to what we're used to seeing. However, there are frogs that produce froglets or basically miniature versions of the adults that hatch out of the eggs. It requires a larger yolk, fewer eggs are produced as a consequence. And so there's trade-offs to different reproductive strategies. You know, our Missouri frogs all produce an intermediate life stage, an intermediate life stage between egg and juvenile, called a larva or a tadpole in the case of frogs. And we probably have all seen some tadpoles at one time or another in our lives. Their body form looks nothing like the adult form. So really what these are free living embryos. Essentially, the yolk has been depleted, but they still need to develop. And so they have to be able to survive on their own. They have to be able to find food on their own. And so this body form is fairly consistent amongst, you know, anurans, frogs that have a tadpole life stage. A couple interesting things: they have gills. Tadpole gills are internal, so the mouth is where water would enter the pharynx. Gills are adjacent to the pharynx. The water would pass over the gill membranes and then out this opening called the spiracle. So there's this unidirectional flow of water across the gills. Hind legs tend to erupt first, meaning you're going to see hind legs on a tadpole prior to the forelimbs, but the forelimbs are developing at the same time the hind limbs are. However, they're covered by an operculum or a piece of soft tissue. So they're developing internally as the hind legs have already emerged externally. And then eventually, once frogs reach or the tadpoles reach a certain stage of development, they'll begin their full transformation into, you know, an adult body form. Now, the reproductive organs aren't mature at this point once they undergo metamorphosis, but the body form is complete. The tissues are recycled, resorbed, and sometimes digested for energy to help with the process of metamorphosis. So they lose the tail, you have the limbs develop, the internal gills are lost. The mouth parts of tadpoles are all keratinized, and so these little grooves or the lips around the mouth are lined with little ridges of keratin. And the significance of this is that if we're in the forest and we're trying to identify tadpoles, most of the dichotomous keys that you might pick up are going to ask you to look at the morphology or the structure of the mouth parts. And so this is species specific, like the layout of the labial teeth or the keratinized rows is consistent within a species, but differs between species. And then also the tadpoles have a lateral line system, which is the same type of sensory system that fish use to detect vibrations in the water, that they use to school, that they use to navigate in the dark, that they use to avoid or detect predators. And so the tadpoles have a lateral line system, which is a holdover from, you know, this is an ancestral characteristic, a holdover that was inherited from fishes. Salamanders in Missouri are a little bit more diverse in terms of reproductive modes. And so this is a plethodontid. This is a cave salamander. Some of our salamanders deposit egg masses in the water, and eggs develop in standing water. Other salamanders in Missouri actually deposit eggs in a moist microenvironment, and then the embryos develop into miniature versions of the adult prior to hatching. And our plethodontids are a great example of a family that exhibits this type of behavior. They do produce like aquatic larvae also, so there's quite a bit of variation within the family. But in terms of an example of one that has direct developing offspring, our plethodontids are a good representative. Now, not only is this just a cool photo, and so this is one of the salamanders that we get to sometimes observe while we're at Rice Biological Station. Cave salamanders occupy caves, but they also occupy the twilight zone on the edges of caves. So you don't have to go spelunking or explore a cave and disturb, you know, the habitat inside the cave to see a cave salamander, because they do live on the edges or the periphery of caves, kind of in the moist microenvironment. Now, this is a male cave salamander. I know it's a male cave salamander because we see this little extension coming off of its nostrils. So if we zoom in a little bit, you can see it more clearly. Salamanders communicate with pheromones extensively. So they produce pheromones. Their courtship behavior is a little bit more intricate and complicated than frogs in terms of when it comes time to produce a spermatophore, when it comes time for a female to accept the spermatophore and fertilize eggs internally. It takes a little bit of coaxing or convincing from the male salamander. And so they produce pheromones, they detect pheromones. Pheromones are sensed inside the nasal cavity in a recess called the vomer nasal organ. Our nasal cavities detect the airborne molecules, things that stay suspended in the air column, a little bit smaller, a little bit lighter, whereas the heavy pheromones tend to kind of sink to the substrate. And so they pick up those pheromones with this extension. It's called the cirri. And then there is a groove that runs from the upper lip in our plethodontids to the nasal cavity. And then the pheromones or the molecules that have been transported via capillary action and diffusion up the nasolabial groove are deposited in the vomer nasal organ. And so that's an important method of intrasocial communication between salamanders within a species. And plethodontids all have a nasolabial groove, and then in the breeding season, the male's nasolabial groove develops that extension, or a cirri. Salamanders, the ancestral salamanders I mentioned at the beginning of tonight's talk, the cryptobranchids, the sirens, the hynobiids, they practice external fertilization. So females deposit eggs in the environment and males spread their sperm over those eggs. Eggs are fertilized in situ. But all other salamanders practice internal fertilization without an intermittent organ possessed by males. And so how is sperm delivered internally to a female's reproductive tract without an intermittent organ? Males produce little sperm capsules called spermatophores. These spermatophores are deposited in the environment by the male, and then the female absorbs or takes the spermatophore into her cloaca and fertilizes eggs internally. So the sperm cap kind of rests on a stalk. This is a nice and neat diagram from one of the textbooks I teach out of here on campus. But if we're looking at the spermatophores and if we're in the forest during a breeding event and we see kind of little white, kind of looks like a melted marshmallow on a little, gelatinous pad, that's a spermatophore. And so these are spotted salamander spermatophores. They deposit them not only on dry land in kind of the moist microenvironment, the leaf litter underneath logs, in the substrate, but they also will deposit spermatophores in aquatic habitats. And so at the bottom of an ephemeral pool, you might see, a series of spermatophores, and females will take spermatophores into their cloaca on land and in water. One of the what I think is one of the coolest aspects of salamander reproduction is that females are capable of sperm storage. This is something that was first discovered in salamanders, and then as we start studying other vertebrate groups and looking at their reproductive anatomy under a microscope, we start to realize that other vertebrate groups are capable of sperm storage, but it was first described in salamanders. And so here is our cloaca, this internal sinus. Here's the vent. And then inside the cloaca is where fertilization of the eggs takes place. But offshoots of the cloaca are called spermathecae. And inside these spermathecae, sperm can be stored and nourished so that females can either store sperm from a fit male, and maybe environmental conditions aren't as conducive to oviposition or depositing her eggs in the environment as they should be for survival of the eggs. And so she can store sperm from a fit male, to the following breeding season, or she can produce clutches of eggs with multiple paternity. Males can produce all kinds of spermatophores for multiple females and spread his genetics, around a little bit in the next generation. Well, females can do that also, so they can generate some genetic diversity in their offspring by accepting spermatophores from multiple males and then fertilizing the sperm from those multiple males, or using the sperm from those multiple males to fertilize a single clutch of eggs. So if we're looking at some actual slides of cloacal tissue from a salamander with sperm present. So we're looking at the body of the cloaca, and then these ducts or chambers that we see in the background are the spermathecae. So Dave Seaver was a herpetologist in Louisiana. He did a lot of histology, and a lot of credit goes to him for kind of shining a light on this aspect of their reproductive biology. And then a little bit closer view inside one of the spermathecae, and you can see the head of the sperm cells embedded in the wall of the spermathecae, the tails extending. And so the environment inside the spermathecae is just right to keep the sperm healthy and nourished until it's time for fertilization. So that's a really cool aspect of their reproductive biology that I don't think a lot of people realize. Some of our salamanders produce gilled larvae. So this is a grotto salamander from Onondaga Cave State Park. And the body form of the aquatic salamander larvae, doesn't differ drastically from the adult form, kind of, not as much anyways as the tadpoles do from, the adult form in frogs, but we can see external gills just behind the head. The tail is somewhat paddle shaped. And so this is an example of a species of plethodonid that produces aquatic larvae that have to spend a time of their life growing before metamorphosis and assuming that adult body form. Some salamanders, like our sirens, retain their larval characteristics through sexual maturity and throughout their life. And so we call these types of organisms pedomorphic, the retention of larval forms or morphologies. And the gills, the external gills, which we can see clearly here on this siren, are an example of pedomorphosis in salamanders. And so sirens are another one of these ancestral clades, along with the cryptobranchids, that practice external fertilization. They are permanently aquatic, and gas is exchanged in these gill filaments. And this siren is from Duck Creek Conservation Area down in the Boot Heel. Keith Cordell, who recently passed away, a terrific biologist for the Missouri Department of Conservation, hosted a couple of our classes, and he was kind enough to set out some hoop nets and some minnow traps, and our students got to experience some of these specialist species that we don't get to observe throughout the year. So just a shout out to Keith. He is a terrific person. Here is a tiger salamander with external gills. Some of our ambystomatids will forego metamorphosis, retain larval characteristics, but become sexually mature and reproduce as pedomorphs. And so, and this is all determined by the environment, competition within the pool of water, competition outside the pool, environmental conditions outside the pool, all dictate the kind of the phenotypic plasticity we see in some of our mole salamanders. So not all salamanders can forego metamorphosis, just in special cases. The mole salamander here in Missouri can do it. The tiger salamander can do it. Now, this is a western tiger salamander. This is another image from our field biology trip, but it's the best one I have of a sexually mature tiger salamander retaining the external gills. And so that's what these are, these branches that come off the back of their head, and then the membranes that hang below them are gills. And so in the case of the siren, this is their life history. There is no metamorphosis. Same thing with axolotls, which were a popular ambistomatid salamander in the pet trade. Their pedomorphosis is obligatory, whereas the tiger salamanders, the mole salamanders we have in Missouri, are facultative, meaning that it can either happen or it can't happen, but it's determined by environmental factors. And so that's a really fascinating aspect of their ecology. This is a slimy salamander, a western slimy salamander, probably one of the most charismatic slimy salamanders that I've ever seen. A lot of white pigment on the body. It's usually not that white. Most of the body is black with small flecks of white, but this one just happened to have this great white, really striking color pattern. That's one of the, I think, the toughest things about becoming a naturalist, about becoming a biologist and confident in your ability to identify organisms, is that we have to recognize variation within a species, but then we also have to be able to recognize variation that differentiates two species from one another. And that's one of the hardest things that we have to do in order to understand what we're looking at in the field. And I see my students at Missouri Valley struggling with it, but eventually you can get it with repetition, with just spending more time outdoors. You start to see different examples of the same thing, and then you start to realize how the variation in one species is actually different than something that you might think is similar in another species. And so just a lot of white on this slimy salamander, a little bit different than normal. The reason why I'm talking about slimy salamanders, though, is because they are a type of salamander that produces eggs that hatch as miniature versions of the adult. So my advisor in graduate school at Arkansas State is named Stan Troth, probably one of the most prolific, publishing herpetologists in recent history. And he had a graduate student, Joe Milanovich, studying slimy salamanders in the Ouachita Mountains in the Ozarks. And what they figured out, what's unique, we already knew that they, are direct developers, hatch out as miniature versions of the adult. But what Stan Troth and Joe Milanovich discovered was that females were returning to the exact same place to deposit eggs year after year. So there was nest site fidelity taking place within this population of slimy salamanders. We don't know if that happens everywhere, but it was definitely happening within this population of salamanders. And this was so extraordinary that David Attenborough came to Arkansas and filmed a segment of his Life in Cold Blood documentary series on amphibians and reptiles on the western slimy salamander in Arkansas. So this is a photo of one of the females in the mine shaft in Arkansas. Same mine shaft, but her clutch of eggs is hatched, and you can see the juvenile salamanders. Their little belly is still full with some yolk, and so they have a little bit of energy to get by on until they can start finding food on their own. And they do nest guard. So there are examples of parental care in amphibians. This ancestral group of vertebrates. Here's the hellbender. This is a healthy hellbender. Hellbenders are an organism that I worked with for several years while I was at Arkansas State, have tried to stay active in the hellbender community in terms of what's happening out there. But, in terms of the salamander, the amphibian diversity that we have in Missouri, if there was one species that is recognizable worldwide, if we were talking to a herpetologist in Germany, they would know about the hellbender. They would be excited to learn about the hellbender. And so it's something that as people from Missouri, we should be proud of. When we think about what makes Missouri special, this is an example of something that makes Missouri special. The Ozark hellbender is endemic to southern Missouri and northern Arkansas. It's found nowhere else on Earth besides that little pocket of Ozark habitat. And unfortunately, its range has constricted over the last 20 years. It's gotten smaller because of some of the threats facing amphibians, some of the threats facing hellbenders specifically. So this is a healthy hellbender. You see all of its toes. I am going to show you some images that are relatively graphic from one of my publications that was detailing abnormalities in Arkansas hellbenders. The photos that I'm going to show you are consistent with hellbender abnormalities in other parts of its range, including Missouri and including the eastern hellbender and parts of Appalachia. So just be fair warned, I am going to show you some gross abnormality photos, but I like starting with this one because this is a healthy male hellbender, about as healthy as you could imagine. Hellbenders have really wrinkly skin, extra folds of skin because they breathe through their skin. They're permanently aquatic. They do not have gills as adults, and they don't come to the surface to take breaths of air. Even though they have lungs, they're not really functional. They're not utilizing their lungs. They're relying primarily on their skin for gas exchange, and having extra skin, wrinkly skin, increases the surface area for diffusion. Hellbenders in Missouri and Arkansas occupy fairly large rivers. Relative to the eastern hellbender, relative to the Andreas in Japan and China. So this is a hellbender river in the southern Ozarks, and it's a fairly wide river. Hellbender habitat, or hellbenders live under large rocks at the bottom of rivers, and they're habitat specialists. That's one of the issues with their decline, is that their habitat is being degraded through riparian zones, shrinking, increased erosion of the bank, sedimentation and siltation of the microhabitat, kind of covering up all the little interstitial spaces that the adults live in, and as far as we know, the larvae also. Now, the Chinese giant, so hellbenders belong to the family Cryptobranchidae. This is a Chinese giant salamander. This is Andreas davidianus. It almost looks like it's walking on land, but you can see some small minnows swimming next to it. So it's underwater, same body shape as our hellbenders here in Missouri. However, the Andreas gets much bigger, close to 1.5 meters, 2 meters in total length in some cases. The world's largest amphibian. But if we look at the stream that Andreas was photographed in, you can see how small it is, and there's not a strong current. The stream is kind of defined by small pools with a little bit of flowing water connecting each pool. And so that was something that struck me, when I'm thinking about comparisons between our hellbenders, in the Ozarks versus the Andreas in Japan and China. The size difference between the species, but then also differences in their habitat, I thought was interesting. This is Andreas japonicus. This is the Japanese giant salamander. Again, larger than our hellbenders. Hellbenders get to about 60 centimeters in total length, whereas our Andreas in Japan and China get to 1.5 meters, in some cases close to 2 meters. And this is a healthy, gravid female. She's about to deposit some eggs, hopefully for a male to fertilize. And the stream that Andreas was photographed from. And so again, a much smaller stream than the rivers that we find our hellbenders in. I'm not saying that we can't find like our Ozark hellbenders in small streams. I'm just saying that's not typical of hellbenders today. They are confined to larger rivers than that. Hellbenders, because the water is a little bit deeper, you got to scuba dive for hellbenders. So that presents a different, problem in terms of how we go about studying them, as opposed to, waiting around in shallow water and being able to see the animal, walking about at night, and then you could, capture the animal relatively easy. Hellbenders, it's a little bit different. In Missouri and Arkansas, the water's deeper, and it requires a little bit more of an effort to survey for them. The person on the left, this is Ben Wheeler. Ben is a professor at Missouri State West Plains in southern Missouri, one of my mentors in terms of becoming a hellbender biologist. And we're diving in the wintertime. So we were doing work, year round, hence the dry suits. And then just a little bit more credit to a few people. This is Stan Troth. This was my advisor. Here's Ben Wheeler. Kelly Irwin on the forefront of hellbender conservation in Arkansas. He's the was the Arkansas Game and Fish state herpetologist. This is Max Nickerson, a hellbender OG. has been studying hellbenders from the 1970s, and then a couple biologists from the university in Rolla, Missouri. Now, in terms of conservation threats or threats to hellbender populations in Missouri, habitat degradation, destruction is consistent with hellbenders amongst all other amphibians. That's always a conservation concern. Now, wildlife diseases are also one of these kind of emerging threats in terms of amphibian populations globally. One of the infectious diseases, one of the pathogenic diseases that has decimated amphibian populations globally recently is a pathogenic fungus called chytrid, or Batrachochytrium dendrobatitis is the scientific name, but essentially chytrid fungus is a parasitic fungus that feeds off of keratin in the epidermal layers of an amphibian. And as the host is infected with the zoospores, which are these motile reproductive parts of a fungus that can actually swim through the water, so they can actually move. They're motile, they move through the water, and that's one of the reasons why amphibians are so susceptible, because one amphibian that's infected with chytrid can spread the infection to other amphibians without actually coming in direct contact with the other amphibian. So if they're just sharing the same water body. So these are motile zoospores that have a flagellum, move through the water, and infect a host's epidermis, and then begins feeding off of the epidermis. And one of the reactions that the host has to the infection is actually to thicken the epidermis to produce more keratin. And so that is induced by the infection. And we're still figuring out the pathophysiology of the disease, and we're still learning about how it impacts different amphibian groups differently. So looking inside the mouth of a hellbender, you see a fungal infection. So this is, we were, Ben Wheeler and I, Stan Troth, were doing hellbender work together in the early 2000s. Chytrid was first described formally by science in 1998 or 1999. And so it was just a couple years after the first documented described case of chytrid was published, that we were observing these abnormalities in hellbenders. And we really weren't sure what we were observing. We just noticed that the hellbenders in the Spring River specifically exhibited high rates of abnormalities. And these abnormalities were not intrinsic or part of the organism's life history, meaning they weren't bite marks from males sparring with one another. These were something that were extrinsic, something that was external to the organism's natural biology. Twofold here in terms of the photograph. You have a large cut wound on the back of the hellbender. That was caused by, a canoe running over the top of the rock, or maybe someone fishing standing on top of the rock. And so not only is habitat being disturbed or destroyed by siltation, erosion in the from the destabilized banks, but if they're occupying shallow water and it's a spot where a lot of canoe traffic passes over, the hellbenders are at risk of being crushed or maimed. Or if someone's standing on the rock to fish and the rock wobbles a little bit, then the hellbender could be hurt. So we that's what's going on here in terms of this large cut. But then we also have a fungal infection. One of the kind of the consistent abnormalities that biologists see when they are studying hellbenders are kind of mangled toes and feet. So the chytrid attacks keratin, or feeds off of keratin, and that's why it embeds itself in the epidermis. That's where it remains. That's its energy source. Amphibians, hellbenders, have a thin epidermal layer, but there are portions of the body that are a little bit thicker with keratin, including their toe pads, and that's also where the fungus lives. And so the fungus is in the substrate. So as the hellbenders are walking along the bottom of the river, their toes are coming in contact with the zoospores, but then also that's where there's just a little bit thicker layer of keratin, and the chytrid tends to attack the toes. And so you see various stages of this infection within the toes, where this one has lost a toe, process of losing another toe. We would see necrotic limbs develop. So after the infection has progressed below the skin, sometimes you see the actual pad of the foot kind of being sloughed off and all the way to the bone in this case. So you can see these little circles in the interior of the palm of their foot. The toes are eventually lost completely to the disease, and a stump remains, and sometimes the stump heals, sometimes it doesn't. Sometimes the disease can be lethal if there's an infection that follows the open wound. But in a lot of cases, the hellbenders tend to clear the infection, and the wounds actually close. And hellbenders will live a long time without actual feet, so they can get by just on little stumps like this. A couple more, pretty severe examples of skin infection. This particular hellbender has sloughed off the area of the skin that was infected with the fungus. See the disease, though, progressing on its hind limbs. On the chin of this hellbender, the skin has been sloughed off. So this was stuff we were observing, in the early 2000s. And we were just doing some basic descriptions of the abnormalities, quantifying the abnormality rate. We were comparing the abnormality rate that we were observing in the Spring River to a healthier population of hellbenders, and then also to hellbenders that were collected in the 1970s and had been stored in the Milwaukee Public Museum. And what we found was that the hellbenders in the Spring River had an abnormality rate over 90%. And unfortunately, this is one of our river systems in the Ozarks that we kind of view as ecologically extinct in terms of hellbenders in the Spring River. So they it seems like have been impacted by influences of decline more than other hellbender populations. But the other hellbenders are still facing, imminent threats that could lead to extinction. And so, if we're looking at this image of the spotted salamander, just real briefly before I get on to kind of the conservation efforts that arose, or hellbender conservation efforts that arose in the early 2000s, we can see this spotted salamander. This is another Missouri salamander, and on its back, this little gray patch of skin. And that is a fungal infection. That's a chytrid infection on the back of that spotted salamander. And then also, if we're thinking another, a tissue slide that's of a frog that's been infected with chytrid, the blue portion of the bracket represents the typical thickness of the stratum corneum, that keratinized layer of the epidermis, and then after infection, so you can see the zoosporangium, the reproductive cell containing structures, embedded in the epidermis, and the epidermis has thickened. And so as the epidermis thickens, osmoregulation is disrupted, respiration is disrupted, and then those symptoms impact other organ systems, and ultimately, death caused by chytrid is not from the actual fungus itself. It's caused by the disruption that the fungus causes in some other aspect of their physiology, beyond straight respiration or osmoregulation. So in the early 2000s, a group of people came together and formed the Ozark Hellbender Working Group, and it had already formed when I came to Arkansas State. So I got to join in the beginnings of this working group. And the working group was made up of concerned scientists, like Ben Wheeler and Stan Troth and Kelly Irwin and Jeff Brigler, both representatives of their state wildlife agencies, Jeff here in Missouri, Kelly in Arkansas, fish and wildlife biologists from both regions. And so Missouri and Arkansas are not part of the same region in terms of how the Fish and Wildlife Service manages wildlife resources. And so it's two different regions, but Arkansas contains Ozark hellbenders, Missouri containing Ozark or has Ozark hellbenders. And so there has to be collaboration, across political boundaries, between different types of agencies, whether it be Fish and Wildlife Service or the Department of Conservation in Missouri. Academicians like Stan Troth, Hugh Wang, biologist from the University of Missouri in Rolla, Mauricio Solis, Ron Gellner from the St. Louis Zoo. I'm going to talk a little bit about the St. Louis Zoo here in a moment. But anyways, a group of people, concerned scientists, biologists, that came together to try to address the things that we were observing of hellbenders in the field. And so the abnormalities are concerning, the evidence of decline, the shift towards larger size classes through time is a concern. So to address the problem, we came together and developed a conservation strategy. And any sort of endangered species conservation strategy really has three prongs. We need to figure out what's going on with the species, identify causes of the decline, what are the threats. In some cases, extreme cases, we need to bring individuals from the wild into captivity, try to captively propagate them, breed them in captivity to supplement the wild populations. Outreach has to happen, and then we need to establish some sort of habitat protections. And those were the goals of this group. Ultimately, at the time that this photograph was taken, the Ozark hellbender was a candidate species for listing for protection under the Endangered Species Act. It didn't have federal protection. So that was also one of our goals, beyond captive propagation, but beyond outreach, beyond habitat conservation. We were trying to get a little bit more federal protection, money geared towards hellbender conservation. And so eventually, our efforts came to fruition. So in 2010, the Fish and Wildlife Service listed the Ozark hellbender as an endangered species federally. It had already been listed as endangered in Missouri and Arkansas prior to that, but without the federal protection, the state protection had its limits in terms of money that's available for conservation. Some of our projects that were the result of this working group. So anyways, this is just a quick snapshot of the official listing of the hellbender, which was a product of that working group. Now the St. Louis Zoo is doing extraordinary things when it comes to hellbender conservation. While we were at Arkansas State, we were trying to captively propagate hellbenders at the Mammoth Spring National Fish Hatchery in Mammoth Spring, Arkansas, with no luck. Other zoos around the country were trying to captively propagate eastern hellbenders. The St. Louis Zoo is working on the Ozark hellbender, but nobody could really get the hellbenders to breed in captivity until the St. Louis Zoo did it. So the St. Louis Zoo was the first organization, the first people that were able to captively propagate the hellbender, and it's a lifeline for the species. It's a lifeline to sustaining them indefinitely into the future. So without captive propagation efforts, I'm not really sure where the hellbender would be at today in Missouri. But just in a few short years, the Missouri Department of Conservation is releasing captively propagated hellbenders back into the wild, and in some cases into stretches of river that hellbenders haven't been in a long time. The hellbenders lay two strings of egg. One string is produced by each ovary. This is a clutch of unfertilized eggs from Arkansas. Then we have some fertilized eggs with developing embryos from the St. Louis Zoo. You can see their yellow belly. And then, a couple hellbenders shortly, post-hatch, their external gills. They still have a little bit of energy in that yolk sac. Hellbenders retain their gills for a little bit over a year, eventually undergoing metamorphosis, assuming an adult body form. And so this is the smallest hellbender that I ever caught. And it was probably the biggest thrill in terms of hellbender research that I ever had, also because it was like the first positive sign that there was a successful recruitment event in one of the rivers that we were working in. And so this was just a little bit of light in a fairly dark tunnel when you're constantly exposed to the abnormalities and the older age classes that we routinely caught. To catch a young individual, a subadult, was a really encouraging experience. I said I would mention Ron Gellner. Ron Gellner was a biologist at the St. Louis Zoo. He spearheaded their captive propagation effort at the zoo. This is an image of Ron at one of our Ozark Hellbender working group meetings. And Ron has since passed away, but the Hellbender captive propagation facility at the St. Louis Zoo is named in his honor. I've been fortunate enough to bring students to the St. Louis Zoo, and they've shown us the kind of the inner workings of their captive propagation facility. It was an extraordinary amount of effort and resources that went into this project for the St. Louis Zoo. But I really have to applaud zoos because historically zoos would take animals from the wild, put them on display without really any thought of the impact that was having on the wild populations. But anymore, zoos are at the forefront of a lot of conservation efforts of endangered species with their captive propagation projects, and they're just the best equipped to do this type of work. And so they are partnered with the Missouri Department of Conservation, and the Missouri Department of Conservation is doing a fantastic job managing hellbenders, which were threatened with imminent extinction in Missouri until people began, really getting involved and promoting their conservation. And so I know I probably went a little bit long. I appreciate your attention. Quick shot of some of the resources I used tonight in case you want to go back and try to find them for yourselves. But other than that, I'm happy to hand it back over to Dana and Ethan and answer any questions that you guys might have.
Great, great. Thank you. Well, if you stop share there so we can all see you. It looks like you got a lot of support there in the chat. We do have some good questions, but you said you ran a little long there.
Yeah.
But I think it was good too. Um, your explanations and the depth of your passion melded with your science and your teaching ability really is a good testament for what good teaching's about, and I think we all benefit from it. If we sit back and think about how that is extended through all the students and people you've interacted with, it's a huge impact. So I think teachers are often overlooked, professors are often overlooked, just like these hellbenders and these other herpafaunas. And I'm glad that you had some time here to talk about these really important things.
I appreciate that, Ethan. In terms of how we relate to other people, I think as a scientific community, sometimes there's just some kind of built-in roadblocks in terms of trying to communicate the importance of what we're doing to the general public. And so in terms of how I teach, I just try to put myself in the shoes of a student or in the shoes of the person that I'm talking with. And then also provide context. I think that's important for people. And so explain why something is interesting or why something's important goes a long ways in terms of helping that person appreciate what you're describing. And it doesn't need to be, overly complicated. I think you can learn a lot, keeping things fairly simple. So.
Well, with that said, we do have some questions in the Q&A and the chat, some really good questions. I don't know if you're able to access those in the Q&A through the Q&A feature, but we can also kind of summarize them as well.
I'm old school. I'd appreciate you guys just asking me the questions, and then I'll answer them at the best of my ability.
I think like the main ones really have to do with do we have reasons to expect hellbenders to eventually develop immunity to chytrid and
Well,
other?
and that's a good question. So hellbenders are an example of an amphibian that can clear the infection. Now, in terms of are we ever going to have chytrid-free habitat? I don't know if that's possible at this point in time. So, chytrid, like I said, was first described, and published on in the late 1990s. But then, as we go back and examine museum specimens, we're finding specimens in South Africa from the 1930s that exhibit, a chytrid infection. The geneticists have sequenced it, and its origin is in the Korean Peninsula. And we're not quite sure how it has spread throughout the world, maybe through the pet trade, but it's such a small world now and people go everywhere. It's easy for things to be transferred from one point to another. But chytrid was in amphibian populations well before the 1990s. And so, like I said, we have museum records going back to the 30s. There are some species that are completely decimated by chytrid. And so that's how we first noticed it, is that you have these populations of frogs in Australia that are just disappearing, or in Panama, just completely disappearing. That raised alarms, and we start doing investigations, and we say, okay, we have this pathogenic fungus. Again, some species are highly susceptible. Other species are somewhat tolerant. Some species tend to clear it. And then some individuals within populations seem to be immune to it. And so I guess there would be hope that even if there's chytrid in the environment, maybe, we have individuals that survive the infection and pass that immunity on to their offspring, and we might develop some sort of natural immunity to it with time. But right now, it's of major concern to biologists globally, because again, some of the species are highly susceptible to it. And there's another species of pathogenic fungus that is also highly lethal to amphibians, but it's right now confined to Europe and Asia. We haven't documented any of the cases in the United States or North America. So chytrid is sometimes abbreviated BD, and then the alternative one in Europe is called B-Sal. It's the same genus, Batrachochytrium, but it's just a different specific epithet, but same impact on
I'd
amphibians.
love to take a deep dive into this and a bunch of other questions at some point. Of course, later on we can also follow up. I don't think we have time to address all these questions, but we can send these questions to Professor Hiler, and he can help try to answer those to the best of his ability. When you're thinking about chytrid, and things that would exacerbate situations where it might be more prolific in certain areas. I think about the Spring River, where they recently added 13 million more chickens in that
Mm-hmm.
geography, and all that's going to end up there. So you're affecting things like excessive nutrients, of course, creating situations for algae in general, as well as other toxic effects and
And
other
the
aspects of
eutrophication,
yeah,
promoting the algal growth, that traps sediment that's being carried down the river, and then that sediment
turbidity.
settles out on the microhabitat. And so that was something I didn't talk about in terms of a threat. Yeah, so you're right on there, Ethan.
There's the people are really interested in things like the disease immunity and adaptation aspects. They're really interested in life history and development aspects. They're very interested in the reproduction and the ID questions, the toxins and the possibility for medical use. In recent
Mhm.
news, Alexei Navalny evidently succumbed to, being assassinated basically using poison. So is there a positive use of those toxins out there?
That's a good question. I'm not sure off the top of my head, but all of our medicines have some, biological origin. And so whether we're talking about natural chemicals that plants produce, or, chemicals that we find within different types of venom or poison. But, you have to study these things in order to find out if there's a practical application. And sometimes it's really hard to receive funding to study, salamander poison if you don't know if there's going to be a reward in terms of a practical application at the end of the rainbow. But it's worthwhile because we do benefit from these types of research projects that make discoveries that we didn't expect. And so that's why it's a discovery. It was kind of unexpected and a surprise, and it came about because we were doing research on salamander poison. And so I'm not quite sure if there is an application right now, but like anything, science is worthwhile, because it's a process of discovery or investigation, but everything we know is a product of scientific investigation. We just keep building upon our body of knowledge with new discoveries generated through science. And so it's a worthwhile endeavor, especially in this day and age.
I think with the combination of science and education, that sort of reminds me of those things like ecosystem services or the value of these particularly overlooked species. Um, we haven't yet figured out a way to put a real value on that, but I think we all can intuit it inherently how
Yep.
important those things are.
Mm-hmm.
Well, everybody, we've kept you quite long. I want to thank Professor Hiler here. We have 109 people still staying on for this.
Some of them are my students, and they're kind of used to me going long. And so shout out to the Missouri Valley students that they got an extra dose of Hiler today. But I usually don't have a problem eating up 50 minutes. So I appreciate everybody's patience, everybody's attention, everything. Thank you.
If you don't mind, we will take questions and comments from the chat and synthesize them, aggregate them, and maybe send them to you, and maybe generate a follow-up email to people, because there's just a lot of interest in Waylon. As you know, this is not really a topic that a lot of people know a lot about. So
I'm happy to. So I appreciate the opportunity. I felt like I was a little bit rough on the start just because this isn't a typical format for me. So I apologize if I came off a little nervous at the beginning. So I was, and so
then we'll have to round you up for an in-person event sometime.
Yeah. That's right. And we have it recorded for posterity that you were nervous giving a presentation.
Yes.
All right.
All right. Well, good night, everyone. Thank you.
Thank you, everybody.